So, all right, afternoon, everybody. I'm Paul Orchard. I'm a pediatrician and trained in hematology, oncology, blood marrow transplant. So I've been interested in transplantation and other cellular therapies for diseases like MLD for 30 plus years, something like that. It's a long time. So I think we'll just go ahead and get kind of started. So I'm not going to spend a lot of time talking about MLD because I'm assuming people that are here have some sense of what MLD is kind of about. Is it possible to make that bigger so the people on the Zoom side go away or something? Oh, yes. Yeah, not to get rid of them, but so, and just to kind of put it in context, so there's 7,000 plus rare diseases, and the definition of rare diseases in the US is prevalence of less than 200,000 people. So it's a relatively small proportion of society, but the costs are huge. And there was a group called EveryLife that looked at this in the last couple of years and presented their data, and it's about $1 trillion per year that these diseases cost society. So in terms of where MLD sits, I mean, there's a number of inherited leukodystrophies, lysosomal, peroxisomal. MLD certainly is one of those. So if it's about 1 in 40,000, there's about 4 million kids born in the US a year, so it's about 100 is what the estimate would be. And again, not to spend a lot of time talking about the nuts and bolts of MLD because this is an educated audience, but aryl A is the enzyme that we're talking about. A deficiency of ARSA results in accumulation of sulfatides that damages the myelin both in the brain and in the peripheral nerves. And importantly, there's a number of forms, of different phenotypes, which is kind of terminology we use. And this makes a big difference in terms of what we can do for patients and offer as therapy. So way back when, Liz Neufeld's group, I think in '68, published these experiments on cross correction, which basically was you had cells that were from a patient that had a lysosomal disorder. They accumulated whatever substrates they accumulate. But if you put other cells, even other cells from different lysosomal disorders in the same tissue culture, they will correct each other. So there was a soluble factor that went from one cell that had that opportunity to make enzyme to another cell that needed it. So that was the foundation for considering enzyme replacement therapy, but also blood stem cell transplantation. So the concept here is you got a cell that's missing enzyme, and you can take a blood stem cell from somebody else who's normal in terms of their ability to make enzyme, and you can engraft them with this person's blood cells. The cells will circulate through the body for the life of the patient and serve as a source of enzyme that can then get picked up and taken into that cell and utilized by the cell. So hopefully you can reduce the amount of substrate accumulation that's there. And it's a mannose-6-phosphate mechanism by which those things happen primarily. All these genes, of course, have been cloned, and it's relatively straightforward to make a pharmaceutical-based enzyme. But part of the issue is with the enzyme therapies for the neurologic diseases is these are large molecules, these enzymes are, and they don't penetrate through the blood-brain barrier very well. And so infusing intravenous enzyme doesn't treat neurologic disease very well. The concept, though, with transplantation is that some cells from the donor are going to make their way into the central nervous system over time after transplant. And those cells can also produce enzyme and serve as a source for the cells within the brain that are missing enzyme. So this is the reason for some of these disorders that are lysosomal but have a neurologic manifestation to them that we're still using transplant, even though enzyme therapy is available for many of the other disorders. So there's a fair amount of experience looking at the role of transplant in allogeneic stem cell transplant in MLD, and it's frankly not great. And, you know, there's clear evidence that transplant does not fix this disease. So, I mean, we published this a few years ago, but, and you can fractionate it into the various phenotypes and whatnot as well in terms of their outcomes. But transplant's more toxic than we would like it to be. There's still a 10 to 15% peri-transplant mortality. Patients are in the hospital for a long time. They get high dose chemotherapy. We make them really sick. And so the safety profile of transplant isn't what we'd like by any stretch. We made a lot of progress over a period of last few decades in trying to make it safer. We got better donors and better antifungals and antivirals and these things, so we can kind of tailor make the chemotherapy better than we could historically, but it remains an unsafe procedure. There's not any two ways about it. Plus, it just doesn't work as well as we would like it to work. So that's led to the big interest in opportunities for gene therapy. So there's several different categories of gene therapy, but the ones I kind of wanted to talk about today are ex vivo therapy, meaning you're correcting cells outside the body. And that's a primarily lentiviral based approach at this point. So this has been ongoing for at least 10 years in Milan, where the vector was put together, a lentiviral vector, to express ARSA, and the virus is used to transduce the cells, have them serve as their own source of enzyme. The patients still need to get treated with chemotherapy because we have to eliminate the cells that are producing enzyme. And we have the apheresis procedure is to be able to harvest their own stem cells. So you can get stem cells out of the marrow, that's primarily where they live, but there's opportunities to treat patients with agents like granulocyte colony stimulating factor and Leukodystrophy, and that releases the cells from the bone marrow so you can access them from the blood. You can actually get more cells doing that, and that's primarily what we're using now to get cells for these kinds of purposes. There's a fair amount of data that's been accumulated in Milan on these studies over the period of, again, the last 10, 12 years. And it was enough to get a license in Europe. So MLD is the name of the drug product there, the cell product. And so it's approved in the EU, and a few months ago, I want to say, I don't know, 6 months or so ago, I think, it was approved in the UK as well. There are also in vivo methodologies of using gene therapy, and generally it's adenovirus associated delivery systems. So there are companies working on this also. So that's delivering the virus into the body, actually directly into the spinal fluid. And the idea that those cells or the virus will infect enough cells within the central nervous system and potentially in the peripheral nerves to treat these patients. So I don't think I'm hooked up here. I think I can wander, but so this kind of depicts the apheresis procedure. So again, we have to, we have to establish that a patient is eligible for the therapy, but they get these combinations of drugs, granulocyte colony stimulating factor and Leukodystrophy. They have a good sized line placed as in a IV or a catheter. And essentially the machine will draw blood out of the patient, goes through the machine, it spins out the cells that we want, the rest of the cells go back in. And that's a continuous process that happens over about 4 to 6 hours. And then at the end of the day, we can determine how many cells we've captured that are sufficient. And the patients that we treated, 1 day has been enough, but sometimes people have had to have it done twice. But these are the cells we're looking for. The CD34s, that's a marker of the blood stem cells. And so there's an antibody that's associated with a magnetic bead that you can add to the cells that you have. It will stick to these cells. You can pull those cells out specifically and purify them to 95, 98%. All the other cells go away. And then that's the population of cells that you add the virus to. So the virus, the lentiviruses are HIV-based vectors, and all the HIV components are essentially gone and replaced with the aryl sulfatase A gene and promoters that will help express that. The cells then get frozen, and there's a testing process to show that you have transduced a sufficient number of cells, and they want to make sure that the cells aren't infected and yada, yada, yada. So there's about a 4-week process to clear the cells and say, yes, this is a sufficient blood product to use. So then we get a call that says, you're good to go. Then the patients come back, and then we admit them into the hospital. There's a cocktail of chemotherapy that we use. Busulfan is the primary drug there. The purpose of that, again, is to get rid of their blood producing cells that are not corrected. So when you infuse the cells that are, they will be the predominant cells there, or at least as much as you could do. So the infusions take half an hour. I mean, that piece is simple. But then over time, when those cells start to come in, we can measure the aryl sulfatase activity and how the patients are doing. So this is kind of the platform for the lentiviral-based gene therapy. So just a couple of slides from the data that was presented. Well, so this is a paper that came out in Lancet earlier this year, the Fumagalli paper that documents what their experience has been with the gene therapy there. So, you know, you can get a sense if you're measuring what the motor deterioration in these patients look like and compare it to untreated patients. I mean, the data is pretty dramatic, essentially. You can also present that data in different ways with different phenotypes of the disease, late infantile patients or the juvenile patients, and again, compare them to untreated patients. And so their functional capacity is, again, kind of dramatically different than what you see with the untreated patient population. So this is comparing gene therapy to nothing as opposed to gene therapy to transplant, just to be clear. So I just want to talk a little bit about the process now. So all the patients have been treated on clinical trials with gene therapy have all been treated in Milan. So there has been no trial in the US. And so my understanding of how this kind of materialized is the group in Milan got funding from Italy to develop some gene therapy approaches. This is one they developed. They sold it, I don't know, 6 or 8 years ago or something, to GlaxoSmithKline. And then GlaxoSmithKline decided they didn't want to develop this, and they sold it to Orchard Therapeutics. So Orchard Therapeutics is now the sponsor of this. So they didn't design the trials. The trials were all ongoing when they actually picked up the product. So we've treated a few patients now in Minnesota with MLD, but Orchard Therapeutics is treating these patients on a compassionate use basis. And so they have criteria by which they decide whether or not they think patients are going to benefit. And so that's part of what's depicted here. So if you're asymptomatic with either late infantile disease or juvenile disease, they'll consider you for treatment. Early symptomatic, early juvenile patients can also be considered. They have to have minimal gross motor deterioration, and they have to be cognitively intact as defined by an IQ of greater than 85. So that's difficult because a number of these patients are hard to test, and getting a score of, we've had to turn down a few patients based on those kind of criteria. So essentially what happens is we see a patient we're considering for gene therapy, we do an assessment. There's a kind of a packet that you fill out, a form, giving all the information, all the genotyping, how the patient's doing, time of onset of the disease, all these kinds of things. That gets submitted to a group at Orchard Therapeutics. They have a process by which they review this, and then and that generally doesn't take very long, couple of days. And then they will say, yes, we will offer therapy, or no, sorry. And there's not much negotiation we can do in terms of those things. If we decide that we're going to move forward, if Orchard Therapeutics says we can do that, then the current process has been each patient has been getting their own IND. So we actually have to put a protocol together that's patient specific, with the patient's information and why we think it makes sense to do this as opposed to transplant, for instance. So we have to make a case to the FDA about what we're going to do and why we're doing it. And they have 30 days to review that. So that can be a problem. We're currently trying now to put together a compassionate use protocol, which basically says these are the patients and the characteristics of the patients we will consider to treat. And so hopefully we won't have to do it on a per patient basis. We'll be able to get approval for a category of patients that is accepted by the Orchard Therapeutics guys too. So when that happens, and we haven't heard back from them yet, but when that happens, hopefully we can bypass this step. So, because the FDA will basically say, yes, patients that are eligible for this based on the criteria you put forward, we will let you treat. So we can potentially shorten things by a month. We also have to get financial approval, which is actually a problem too. So if you go back to an insurance company and say, allogeneic transplant, the bill may be $1 million, and the bill for this may be $250,000 because it's an auto transplant versus an allo transplant. You would think that that would make be an important argument for those guys, but they kind of look at the letter of the law in terms of the protocols. And so if the protocol says we don't pay for investigational procedures, then they say no. And so we've had to appeal these things for a couple of patients to try to get them to move forward, but that can slow the process down significantly as well. And Orchard Therapeutics wants to have all those pieces worked out before they will set a date to do the manufacturing. So the manufacturing, they can do 1 kit a week. And so you have to put it on the schedule, and it may be 3-4 weeks out before you can actually get a manufacturing slot. So there's a built-in significant delay there. Once that's approved, though, the process of doing the apheresis is about 7 days with the drugs we talked about, and then actually getting the cells. So what happens from there is, so the cells go to our processing facility. They're shipped to Milan because that's where the engineering takes place. The actual transduction only takes 5 days or a week or something. But again, the quality control piece over and above that to make sure that the product is sufficient is another month. So then they give us a call and tell us that we can move forward. The family would come. We make sure that they still look like they're okay. That only takes really a couple days. And then they get admitted, get chemotherapy, and the cells are infused. But it could take a good month for the cells that we have infused to recover in that patient. And you're not going to get a benefit until you get these cells that are making enzyme to recover. So there's a pretty extensive time lag in this now from when we hear of a patient that might be considered for gene therapy to when they can finish the entire process and actually potentially benefit from it. So this is problematic. We had one patient that had early symptomatic, early juvenile disease who lost a fair amount of ground from the time we first saw her and established her eligibility to the time she actually got to transplant. So, I'm hoping there'll be ways that we can make this better, but at least for now, it remains a problem. I just wanted to talk a little bit about some of the other approaches. So the adeno-associated virus is going to be delivered. So there's a company working on this. There's a couple companies that are doing it for other neurologic diseases. This is also being developed for MLD. But essentially, this virus is delivered actually into the body of the patient. And they're doing it into the cisterna magna, which is kind of a pocket of CSF, a spinal fluid that's in the back of the brain. So you have to have a dedicated, skilled neurosurgeon to do that. There's a lot of blood vessels and whatnot there, and you could hurt somebody. So, but they decided this was the best approach. And then the virus can move through the brain, through the spinal fluid, and then hopefully make enzyme. So that's the approach for the AAV side of things. There was some discussion about how these things may work because this is treating the central nervous system. It's not directly treating the peripheral nervous system. And is enough virus going to actually leak out of the brain? Because that's what seems to happen to some degree in the animals. But do you get enough virus that gets out of the brain and produces enough enzyme in the periphery that you're going to treat the peripheral nerves well? And that will be an important thing to assess. But I think this is a year or two away for MLD. So just for a synopsis here, for now, transplant is standard of care, but as someone who does transplants, it's inadequate for this disease. Diagnosing patients because they're symptomatic is also insufficient because they're progressing. Some of the kids progress quite quickly, and we don't know necessarily for gene therapy, perhaps, but certainly for transplant, nobody recovers anything that they've lost. And so, you're not going to get a better outcome than what the patients look like at the time they were treated. So we need to be able to identify these patients as early as possible. And it's pretty clear that newborn screening, at least to me, newborn screening is the best way to approach this. A good chunk of the patients that were treated in Milan were, if not almost all of them, were second siblings of kids that have already been severely affected by the disease. And that's obviously a small proportion of patients and not something you want to rely on. So, and there are other things, hopefully they're coming down the pipe too that might help us, but newborn screening is going to be key. So I'm going to stop there. I was just talking to Angela Tom, who is with Orchard Therapeutics. I think she had to leave, but they're going to be applying for a license in the US, and probably late this year, they'll start that process, but it may be 18 months or so before they get a license, assuming that they get a license. And the data, I think, obviously looks very good. So I would be surprised if they weren't successful in that, but they aren't asking me. So we'll see where those things go. But in the interim, we're not likely to have, outside of the compassionate use side, anything else to offer but transplant. So we have to do that. So any questions about any of those things? Sure. I doubt it. I've asked the company that a couple of times, and so I think for right now they're going to rely on Milan. At some point, it's hard to imagine that they aren't going to want to do that. I mean, they've got other diseases they want to treat in the pipeline too, and to have to send all the samples to all the cell products to Milan and ship them back can't be optimal. So one would hope that that's going to happen at some point. Again, it's hard to imagine they're not going to, but I don't know what the time frame is going to be. I mean, I think part of the thing with the FDA is if you have another treatment facility, you're going to have to prove that that treatment facility is similar to the treatment facility that is already, in process. So that how much time and effort and money that's going to take to be able to do that, I don't know, but it's not high on their priority list, I don't think, at least for now. Probably not.
Please repeat the questions. We're not hearing them online. We only hear the answers. Thank you
okay.
so much.
Yeah. Sorry. So, yeah, the question there was whether Orchard Therapeutics is thinking about having a manufacturing facility in the US so that the cells wouldn't have to be transported to Milan for the engineering and be transplanted back. So, again, I think it's likely that's going to happen, but probably not in the real near future. So, I mean, one of the other things that's going to be a issue for these kinds of therapies now, and so importantly, Bluebird Bio, which is another lentiviral company, had a couple of things that were far enough advanced that they actually presented things to the FDA formally 2 weeks ago. One was adrenal leukodystrophy, which is another pediatric white matter disease, and the other was thalassemia. But these were day-long meetings where the data of efficacy was presented, the toxicity stuff was discussed, and at the end of the day, they made there was a vote essentially as to whether this FDA advisory committee thought it made sense to consider moving forward towards licensing. And for both of those products, the votes were unanimous, which was very exciting to see. So, and presumptively, if there's a green light for those kinds of therapies, then MLD one would anticipate would be similar enough that hopefully you aren't going to have a ton of problems there. So that was very encouraging. Another set of issues we kind of talk about this that people want, but is what's the cost of these things going to look like? And that's not yet clear. I think it was close to $3 million for product in the UK. So that's kind of daunting. So if it gets licensed, then presumptively the private insurance pieces or groups are going to have to pay for it. But I don't know. The healthcare systems, of course, in Europe are different than they are here in the US. And so if you get approval by the National Health Service in the UK, then everybody's on the same health service. Whereas in the US, that's certainly not the case. So some people are going to have private insurance, and then some people are going to have state insurance, and state insurance tends not to pay very well. And so those issues are going to have to get dealt with too. But we'll see where those things go. So, okay. So, John, do you want to talk for a bit here or something too? I can swap this out.
Okay, I'm Tony Rupar from London, Ontario, Canada, and pleasure here to be at this meeting. Now, I've been interested in metachromatic leukodystrophy for a number of years, and at our scientific medical session yesterday, I presented some a perspective of the sort of research we're doing. So I didn't bring the slides with me because there's full of a lot of detail. But I'll give you an overview as to what we're at, and very much following Dr. Orchard's points is that, of course, the Milan approach is really only works for pre-symptomatic children in the late infantile, the most common form of the disease. And again, that process, by the time you do the infusion of the transduced cells, you're talking several weeks to a couple of months before these cells are actually start to have any therapeutic effect. And if you look at the late infantile MLD is really a very, very aggressive disease, and that's a lot of time in the life of a symptomatic child. So we've been working, looking at direct delivery of viral vectors, including AAV, as Dr. Orchard was discussing. And the advantage there is that you can get expression of the enzyme within about 4 or 5 days of doing the treatment, not 4 or 5 weeks. And now, as the work that's going on now is to, Dr. Orchard talked about injections into the cisterna magna and so on, and there are other strategies as well. And basically is to find what is the most efficacious way to go, and also keeping in mind that although the central nervous system disease is overwhelming in MLD, there's also a peripheral nerve disease too. And we want to make sure we are not forgetting that. So that's one our focus of our research. The other area is approaching, asking the question, are there other therapeutic targets other than the obvious one of putting in a proper copy of the gene? And so we've been spending some time looking at sort of a cellular level as to what else is going on. I mean, we know that fundamental that the individuals are missing a functional aryl sulfatase A. They accumulate the substrate, sulfatide, and then why does this, make them sick? And so we're exploring those details. And there's another, the lysosome, which this is a lysosomal storage disease, is one sort of organelle within the cell. There are other organelles, and particularly mitochondria. Mitochondria are interest. Mitochondria, in a sense, we tend to put everything in it neatly in a basket. So mitochondria have different jobs to do than lysosomes. We describe the lysosomes as sort of the garburator of a cell, breaks down stuff that needs to be broken down. And because it can't break down sulfatide, it accumulates sulfatide, and lysosomes don't work well. Mitochondria, we tend to look at as primarily the source of energy in the cells, but they also get involved in a lot of other physiological function, including cell life. So what some of our research is indicating right now is that there seems to be crosstalk between the lysosomes that are accumulating sulfatide, and the mitochondria in those cells that have also other sorts of jobs to do. And so we're exploring that sort of crosstalk situation, and with the idea of understanding what sort of the underlying processes are, and maybe they can reveal some other targets that we should be looking at to treat. And another area that we're exploring is a inflammatory process too, and looking at this in the context of the peripheral nervous system right now. And again, we're getting some indications that cells that are Schwann cells, so these are sort of the cells that wrap around the axons in the peripheral nervous system to enable them the nerves to work, that they seem to be able to produce inflammatory things we call inflammatory cytokines, in this, which we can imagine is contributing to the destruction of the tissue. So, and it's a different path, but the stimulation seems to be the accumulation of sulfatide within these Schwann cells. So again, that looks that it may have some potential as being another sort of target area that may be helpful to patients. And I really want to echo what Dr. Archer said, is that I can't imagine that anything we come up therapeutically will repair damage that's been done. The most we can hope, I think, reasonably, that we can prevent damage from occurring. And so this brings back the whole concept of early diagnosis. And as Dr. Orchard just said, the Milan protocol is great, but pre-symptomatic children. And the outcomes are really quite exciting there. However, that to have a pre-symptomatic child diagnosed with MLD, other than being a child who is already known to be at risk, it doesn't happen. They come in symptomatic. And when they're symptomatic, that's because the damage has started. So I think it's going to be a few years. I'm optimistic that the newborn screening that Dr. Gelb is doing, developing, I think that will work. I'm an optimist and a strong advocate of newborn screening, not just for MLD, just for lots of diseases. And so I think it will be a few years. It's hard to predict how many years that there will be newborn screening for MLD, that because the process it has to go through. And I'm from Canada, it's the process, the details are different, but basically the timelines are similar. To get something adopted into the newborn screening program takes a few years. So we are a few years away from seeing newborn screening for MLD, but there's optimism here that we will get newborn screening for MLD, and that will be coupled in with some existing and newer therapies that I think should make a considerable difference in outcomes. Did you want to say anything else, Dr. Archer?
Oh, no. So there was Dr. Friedman, I think, was going to be.
Oh, he's not here.
Oh.
Oh,
all right.
Okay.
So, well, no, I mean, I don't really have a lot more to add. I mean, I think in terms of why there's as much variability as there is for some of these diseases, whatever we can learn about the biology, hopefully there'll be other directions that we can kind of attack, because, if there's means by which we can help stabilize these patients while they're getting set up to have a definitive therapy, I think that'd be great. But so accumulating sulfatides, for instance, we think is directly damaging to the myelin, but the inflammation that's associated with it, or the oxidative stress, or some of the other various parameters might be something else that we can target and might help us get better outcomes than we would otherwise get. I mean, so I was in the ALD session this morning too, and we were talking about as we go through bone marrow transplant, for instance, these patients tend to lose a lot of ground in a hurry. And, they may stabilize to some degree afterwards, but it seems the stress of going through the transplant procedure is such that it actually accelerates the disease. And, if there's...
And it's not a benign process.
Well, right. So, I mean, we're giving them, toxic chemotherapy, plus there's inflammatory pieces that are associated with this, and nutritional issues, and they're on a number of other drugs. And so it's a multifactorial issue, but it's a major challenge for us because, like I say, the patient you bring into the transplant service, and when they're hospitalized, they may be at one level, but when they leave the hospital 6 weeks later, they've lost a significant amount of ground, and they tend not to recover it. So, again, if there's other things that we can do to help in stabilization of the disease in the interim, that would be really helpful. So
why don't we open this up to
absolutely.
discussion?
I'll let you.
Okay. Dean, you had maybe your hand up first?
Yeah, can you open your crystal ball and see anything about
You have a better crystal ball. I
Well,
think.
I don't know about that at all. I mean, so, well, so none of the adults have been treated with gene therapy. You know, and presumptively at some point, someone will be willing to do that. But I actually broached it to see if there was interest in compassionate use therapy. And so the bottom line from Orchard Therapeutics is essentially been the criteria that we've had to treat patients on the clinical trials will be the only criteria that we will consider for now. So, I mean, I don't know, maybe down the road, if they get licensed, then maybe there'll be an opportunity to do those kinds of things. But at least for now, it's not kind of on the table. I'm just making it increasingly harder. Oh yeah. So sorry. The questions were crystal ball wise, what might be available for adults with MLD. And so, you know, we were just talking about the gene therapy side, but yeah, I don't know if there's other opportunities. I mean, we've been doing transplant in those patients that even there, it doesn't work very well, frankly. I mean, I think it may slow the disease process a bit, but people still progress. And, you know, I'm seeing someone in a couple of weeks who was, I think, transplanted when he was 17, and from a gross motor standpoint, he's doing pretty well, but from a cognitive standpoint, he's kind of slowly losing some ground. And so, you know, transplant's insufficient. I mean, there may be other opportunities too. So we don't really understand very well how donor-derived cells make their way into the brain. And it's likely a small proportion of cells that come from the donor that will make their way there. So almost all that data is from mice. But so busulfan is a drug that penetrates into the central nervous system pretty well. And the concept is you're actually clearing some of the cells that look like microglia. They're probably not truly microglia, but something that looks like that. You're actually clearing space there for the new donor-derived cells to come in. So there was interesting studies. So if you use treosulfan, which is another kind of related chemotherapy, but doesn't get into the head, you don't see hardly any cells that are from the donor that show up in the brain. And people have done studies too in the mice where if you irradiate half the brain, you get more cells into that side of the brain than you do on the other side. So there probably are things that we can do to influence that. Presumptively, you're just not getting enough enzyme delivery. I mean, maybe that's kind of simplistic, but you're just not getting enough enzyme delivery. And if you're only getting 5% of the cells or the microglia after transplant that are donor derived, and you could double that or quadruple that, then maybe you get better outcomes too. So maybe there's opportunities to do those kinds of things, but it's you know, frankly, from a transplanter's standpoint, I mean, we've been doing these things for 30 plus years and we still don't really know what the heck we're doing, because it's so hard on a clinical basis to know what brain engraftment looks like. I mean, you have to get a piece of the brain to do that.
So probably the donor cells have to find their way into the recipient's bone marrow, you know, big source where the bone marrow is, has been cleared out. And then the cells get there, they colonize, you know, they find a home, they get established, and then these stem cells start differentiating into the cells that eventually, well, some of them will crawl across the blood-brain barrier. So it's a complex process, probably, but we don't fully understand that process.
So on that same thing, what needs to happen for us to get from point A to point B? This therapy, right?
So essentially, the question is, what further information is needed to be able to push things along for treating adults?
Oops.
Crystal ball.
Yeah, well, so I don't know how these things are going to potentially work. I mean, if we get a license, then it kind of depends a little bit what the I shouldn't say we, but if the license is obtained, then exactly what the license is going to look like is not yet clear. I mean, I can't imagine they're going to license it for adults because no adults have been treated. But once it gets to be licensed, then maybe there's an opportunity to do trials on patients that have, you know, adult form of the disease. And maybe if you can generate enough data, you can expand your license or something. It might have to kind of go that direction. So I don't know, I'm speculating really at this point, but it's a similar circumstance for ALD too, because no adults have been treated with ALD either. And so, you know, I've kind of talked to Bluebird about whether it be, if we got approval, would you consider, you know, a small trial to treat some adults? Because the adults don't tolerate chemotherapy as well as the little kids do either. And, you know, so I don't know if that's the direction we might have to go, but yeah, it's not very clear.
I could add a little bit to that, is that there's another lysosomal storage disease, Fabry disease, which is not a primary neurologic disease. The target organs there are the kidney and the heart and other tissues. The clinical trial has gone successfully in treating adult males with Fabry disease. So they would have been aged, well, I think they're generally 30, 40, in the 30s and 40s. So the actual process is very, very, very similar. It was a lentiviral vector that was used on those men. Just exactly the same process that's outlined here for MLD. I think there's one difference, was the conditioning regimen was melphalan was used. Right? And that has the original 5 patients, men who've been through that. They're 5 years out now, and without any adverse effects of the process. It's Fabry disease is clinically a much slower progressive disease, so it's going to take much longer period of time to fully appreciate the clinical impact.
Yeah, so there's no technical reason you couldn't do gene therapy on an adult. I mean, it's the same process. You need more virus because you got a bigger person. So you got somebody that's, you know, 20 kilos versus 80 kilos, then you need, you know, 4 times as much vector per patient treated. So, you know, maybe that influenced what was going on in clinical trials. I don't know, but, you know, it like I say, biologically, well, I mean, the disease looks different, and the outcomes that you'd have to come up with to show that you made a difference in the disease are going to be different, right? I mean, the little kids have primarily motor problems, and the adults primarily have behavioral kind of executive function sorts of problems. So you'd have to target different outcomes, but not to say you couldn't do that. And if the disease is a little more slowly progressive, then it makes it a little harder too, because if you're trying to show that you're bending the curve and, you know, it takes 5 or 7 years to get there, then it just makes the trials harder to run. But you know, it shouldn't be impossible, wouldn't think.
Yeah, the question about cost is really up in the air, because none of these treatments have become, you know, regular standard of care yet. I'll go back to the Fabry disease example. There is a benchmark there. So there is a enzyme replacement therapy that's a standard of care for probably about 15 to 20 years now. And there's also small molecule therapy and so the cost of treating an adult male, like I keep talking about males because it's kind of like adrenal leukodystrophy. Males are, females are much less severely affected. So treating a male for enzyme replacement therapy, depending on the size of the man, is probably running about $400,000, $500,000 per patient per year for the rest of the life. So and if we look at how some companies have priced some of these things out, if we look at spinal muscular atrophy, the cost there has been, I'm being a bit cynical here, has been what will the market bear? So so one might imagine, okay, if a gene therapy is a one and done, and that's our fingers crossed type of situation, then probably the cost will come in for the equal, if at least a few years treatment of the standard of care. So just, we don't know that, but I can sort of imagine doing that. Now, wonder if we should give the people online a chance to ask some questions. I'd like to give a shout out to Dave McIntyre, who heads the Bethany's Hope Foundation that has funded a lot of the research we have done. Are there any questions?
Any questions coming in from chat? Oh, I see. It's just people who might.
Okay.
Yes. No. So historically, there's a couple different groups that will measure aerosolipase activity, but it's almost exclusively in cells and blood. So, I mean, that's a routine thing for us to do. As you go through transplant, you recover your white blood cells. You can get the enzyme assay done on that. And essentially what they do is they spin the cells out and lyse them because the lysosomes are inside the cell. And then they look at what kind of enzyme activity it is. So it measures function. It doesn't measure, how much protein is there. So, I mean, that's quite straightforward. Again, there's a couple of companies that are, groups that do that. The group in Milan developed a enzyme test for spinal fluid. And they've been doing that in their patients. I can't remember how early they've been doing it, but they're the only ones, frankly, the only ones in the world that are doing it as near as I can tell. So there's nobody in the US that's offering that as a routine assay essentially. So I hear the group in Greenwood, South Carolina is actually going to maybe offer it on a research basis because they're looking at it, but that's kind of the level that it is for now. So the blood stuff is pretty easy, and generally by the time you recover a decent white count, you can look at enzyme and enzyme levels. And well, all three of the patients that we've done, at least the first one was higher than normal, and in a couple of them it was 4 or 5 times normal. Whether, what it's going to do over time, I don't know. I mean, all the kids are less than a year out from having gotten treatment. So we'll see where that goes. But I mean, the data from Milan actually looks like the enzyme expression is pretty stable. So we'll have to see. And I don't think there's any way to measure sulfatides in spinal fluid or anything, is there? I mean, that, other biomarkers that might be helpful would be something to consider as well.
The locus where you inject. So you were saying that right? It's the enzyme that crosses the blood-brain barrier, so that that are injected right through the intestinal tract.
Okay. Yeah. So just to clarify that, yeah. So the question is about where to deliver. Now, if you're talking about direct delivery of the enzyme, and that has been done, so one route is intravenously, in MLD. However, that enzyme does not cross into the brain, doesn't cross the blood-brain barrier. So there's no efficacy there at all. Perhaps some of it gets into the peripheral nerve and so on, but So now the other approach, and actually this has been done with purified enzyme, is into the cisterna magna or lumbar. And again, that gets into the brain, but there's a clinical trial that there's actually a device, I think it's said every 2 weeks forever. These patients have to receive that enzyme delivery. And it's questionable whether that has any impact on the peripheral nervous system. Now, if you're using a gene therapy vector, such as AAV, again, that's been discussed into lumbar or cisterna magna, then that will get in, as Dr. Orchard's diagram showed, will get in and potentially treat the brain, the central nervous system, the spinal cord. But there's not much evidence that will get out of the central nervous system and into the peripheral nervous system. So this brings up a discussion then is actually dual treatments. And then some of the research we've been doing with an AAV vector is intravenously and looking at this is mouse work still, and looking at the sciatic nerve, which doing neurosurgery and on a mouse is a challenge. And dissecting, anyways, the sciatic nerve is a big enough nerve you can actually get it out of a mouse and do that. Anyways, so you can get delivery into the intravenous delivery will affect the peripheral nervous system. So of course, this raises the question, what about 2 deliveries? Now, it gets a bit complicated here trying to do 2 treatments simultaneously for the regulatory authorities. And also, and there is an issue with AAV that because most of us will form antibodies or maybe already have antibodies against AAV, and then you may not get, it's not clear whether you get to, 2 or 3 years later, be able to do a repeat dose. There's a number of questions up in the air there.
I know that there was at least one company that was talking about whether to try to treat the peripheral nerve a bit, that it might make sense to do both cisterna magna delivery as well as an LP. So, you give 60-40 or 75-25 or something like that. But I don't know if any of those things are moving forward, but that was, kind of discussed, is that you may effectively treat the brain with those approaches, but not necessarily treat the peripheral nerve as well as you would like. So I don't know. I mean, those approaches are probably going to evolve over time, but so with transplant, the idea is just, you got cells circulating throughout the entire body. They just don't make a ton of enzyme. So that's been the issue. But I mean, we don't have a way with transplant of dialing up the amount of enzyme expression. So gene therapy is a much better approach for that.
Dean?
This might go back.
Okay.
Area of the work while we're doing it, but we talked about not being others might be doing in that space, or is it less some work and it's
Oops. Yeah, so the question was whether or not there may be an ability to restore damaged myelin. And I mean, there are groups that have been working on these kinds of things. So can you get an oligodendrocyte precursor, for instance, and deliver that? And will that localize to where it needs to go and actually restore some of the myelin that's previously been lost? And that'd be really important if we could figure out how to do that. I don't know. If there's anything on the cusp of being tested, I don't know about it, but maybe there's mechanisms to do that. But yeah, unfortunately for almost all these leukodystrophies, whatever damage has been done has pretty much been done. So And, you know, it's been our experience too, especially for ALD, that patients progress functionally and their MRIs don't change very much. So the MRIs are, you know, the best opportunity we've got to look at what's going on in the brain, but it's not the whole story. Because, you know, some of these kids, as we were talking about before, that progress going through transplant, their MRIs really aren't much different before and after transplant, but functionally they're dramatically different. So, you know, maybe there's going to be new opportunities in imaging to help us figure some of those things out as well.
As you're asking that question, I was reminded of MS, which is a far more common disease. And in fact, it's not uncommon for adult patients with, say, Krabbe's disease or to be misdiagnosed as having MS and be followed in MS clinics for a period of time. And oops, I'm not aware of any real progress in MS, which is a much larger patient population. And so basically regenerating the nervous system.
Yeah, not to open a whole nother can of worms either, but we've seen for some of these other diseases when we started doing newborn screening is you start picking up a number of people that you probably wouldn't otherwise have picked up. And so maybe the incidence actually is going to be significantly higher than we thought it was. But especially in the Krabbe literature, you find people that have low galactocerebrosidase levels and you don't know if they're ever going to have a phenotype. And what do you do with those patients and how long do you follow them and what kind of follow-up do you do for them? And so I don't know. I mean, we may end up, if we do a newborn screening for MLD, have the same sorts of issues. Is how are you? So, you know, we're having some ongoing meetings trying to figure out how we might be able to parse these things out. So if you have a patient that you know is going to get sick and by the time they're a year or two old or even going to have juvenile disease, those patients presumptively you would take to an intervention, whether it's going to be gene therapy or something else. But, you know, if you have somebody that you're not sure that they're going to have symptoms until they're maybe 30, what are you going to do with those patients? And, you know, maybe patients you don't know they're going to have symptoms at all. And how are we going to sort that out? And what are we going to do for them? And so that's one could imagine that those issues are going to be something we're going to have to sort out.
Yeah, and that's a really valid point. For example, if you knew somebody who's going to develop, say, neurologic disease when they're 40 years old, you would not go put them through a transplant procedure, the risks and so on, as an infant. That would not make really any sense because who knows what therapies will be available to them, you know, in the future. You just wouldn't run that risk for that as a child.
Yep. So the question was whether or not people that have previously been treated, for instance, with transplant, whether they could undergo gene therapy. And I don't know technically that you couldn't do it. It'd just be kind of a matter of what the license is going to look like and whether insurance is going to pay for it and all these other kinds of things. I think there's some discussion in the Krabbe side as the gene therapies are starting to roll out, whether or not maybe some of the first experience might be to take some of these patients that have previously been transplanted and treat them with gene therapy as one of the initial cohorts and try to find out if, you know, you can get significant benefits in terms of enzyme expression and maybe some functional changes and whatnot too. So I think the door's open to do those things. But the clinical trials that are, you know, have been underway haven't gone there. And so like I said, the AV question has been one where it's not clear that you can retreat people, but you could potentially do that with the lentiviral approach. And certainly we've treated patients that didn't engraft with allogeneic transplant and retransplanted them. So it's a similar kind of thing. You still have to get chemotherapy again and whatnot, which nobody obviously wants to do that, but there's not impossibility that you can do it, certainly.
Okay, anything else we should discuss or could discuss? I don't think we've answered all your questions, but we've done the best we can. Okay.
I'd just say thanks too, and you can find us probably if there's other questions later. So nothing else through the chat or anything here. Okay. All right. Well, thanks everybody for their attention and interest, and again, come find us later if we can help.